Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Photosynthesis in Higher Plants. Today let us discuss the next cycle, that is C4 cycle. This is also another cycle which is happening in addition to normal photosynthetic cycle in certain plants. So normally photosynthesis has got light reaction and dark reaction for process of making carbohydrate. But in plants in temperate region under high temperature, high intensity of light and high oxygen concentration, instead of Rubisco catalyzing carboxylation, it catalyzes oxygenation, and as a result, instead of two PGA molecules, one PGA and one phosphoglycolate will form. PGA will enter into C3 cycle, normal photosynthesis, but phosphoglycolate will undergo C2 cycle, which is actually a wasteful process. No ATP is produced, rather ATP is utilized. Carbon dioxide, instead of being fixed, it is also released out. No glucose is also formed. So it is an unnecessary process. Imagine in temperate region, temperature is very less usually. But tropical plants, they are always exposed to high temperature, high oxygen, high light intensity. So if they all start doing C2 respiration continuously, the overall photosynthetic efficiency will be very less. Productivity will be affected. As a result, the plants in the tropical regions are adapted to another cycle called a C4 cycle. Why C4 cycle? To avoid C2 cycle. So which all plants carry out C4 cycle? Sugarcane, sorghum, maize, millets, etc. These are all grass varieties, mainly monocots. These plants have C4 cycle to protect them from an unnecessary process called a C2 cycle. So if there is C4 cycle or C4 plants, they don't have C2 or photo respiration. It's a cycle to avoid or photo respiration. Now C4 cycle, it is called C4 because the first stable compound formed is a 4-carbon compound called the oxaloacetic acid. So it is called C4 cycle. It is discovered by Hatch and Slack, so it is also called Hatch and Slack cycle. OAA or oxaloacetic acid is a dicarboxylic acid, so it is also called a dicarboxylic acid cycle. Now, how can it avoid C2 respiration? You know that photorespiration or C2 cycle happens in the presence of oxygen, right? Carbon dioxide and oxygen have got the same binding site. So if carbon dioxide is more, carbon dioxide will bind. Oxygen is more, oxygen will bind. So here these plants have got a separate cycle in order to ensure that near Rubisco always carbon dioxide is there. So in the absence of oxygen, can it carry out photorespiration? No. When Rubisco is around, only carbon dioxide is available. So naturally carboxylation will happen, carrying out our dark reaction or C3 cycle. So this is the adaptation, simply avoiding the presence of oxygen near Rubisco. So for this, these plants have a special kind of anatomy, leaf anatomy called a Kranz anatomy. You can see in this picture. Usually we know that in a leaf, there are the mesophyll cells are divided into upper palisade and a lower spongy, but here you can see all mesophyll cells are alike. There is no differentiation into palisade and spongy, first of all. Second, around the vascular bundles you can see bundle sheath cells. Those bundle sheath cells are very large and arranged in a circular manner with a large chloroplast in them. So this gives the appearance of a wreath. Kranz in Latin means wreath. Wreath means, you know, that floral decoration, right, in a circular manner. So this is called, that's why it is called a Kranz anatomy. And the chloroplast inside the bundle sheath cells are agranal. We know granum formed by pile of thylakoids. It is agranal, whereas the mesophyll cells have normal chloroplast with the granal thylakoids. Here this process happens both in mesophyll cell as well as the bundle sheath cell. So two enzymes are involved here because you know that mesophyll cell is exposed to outside environment. There Rubisco is not there, only PEP carboxylase enzyme is there. Whereas Rubisco is kept inside the bundle sheath so that there won't be any oxygen around it. How? We will see that now. So as we discussed, the idea is to keep only oxygen near Rubisco and oxygen away from Rubisco. So for that, the mesophyll cells have the primary carbon dioxide acceptor called a phosphoenol pyruvic acid. So phosphoenol pyruvic acid will combine with the carbon dioxide in the presence of an enzyme, phosphoenol pyruvic acid carboxylase enzyme. So this will catalyze only carboxylation reaction. As a result, a 4-carbon acid called a oxaloacetic acid will be formed. Now this oxaloacetic acid is converted into another 4-carbon acid called the malic acid. Malic acid sometimes will become aspartic acid, but it is a reversible reaction. It will come back as malic acid. Now this malic acid will be shifted to the bundle sheath chloroplast. So up to this, formation of malic acid happened in mesophyll chloroplast in the presence of PEP case enzyme. Now malic acid is shifted to bundle sheath chloroplast. There who is there? Rubisco is there. So the malic acid is 4-carbon. It will undergo decarboxylation, that is removes one carbon dioxide to become a 3-carbon acid called a pyruvic acid. So from 4-carbon malic acid, 1 carbon dioxide goes out means 3-carbon pyruvic acid. So that carbon dioxide released is near the Rubisco. So Rubisco has only carbon dioxide. So this carbon dioxide will bind with the Rubisco, and the Rubisco will catalyze what? The C3 cycle. So it will enter into C3 cycle, and efficient C3 cycle will happen inside the bundle sheath cell. Now this pyruvic acid has nothing to do in the bundle sheath. It will come back to the mesophyll cell. There in the presence of ATP, it will be converted back into phosphoenol pyruvic acid, which again will combine with carbon dioxide, form oxaloacetic acid, malic acid. Malic acid goes to bundle sheath cell, release carbon dioxide. Rubisco will carry out photosynthesis, C3 cycle. Then malic acid is becoming pyruvic acid. It comes back and become PEP. So this reaction ensures that near Rubisco only carbon dioxide is available. So there is no chance of photorespiration. Only C3 cycle will happen. So this additional process to avoid C2 cycle is actually very efficient. C4 plants are highly efficient. Why? Because they avoid C2 respiration. But it is little energetically expensive, because you know that usually a normal C3 cycle uses only 18 ATP molecule, but here the 18 ATP for the C3 cycle needed, but the Hatch-Slag cycle needs another 12 ATP. So total 30 ATP. So it is little energy expenditure than normal cycle, but efficiency is also equally high. Hope you understood the concept very well. If so, please like, share and subscribe to my channel Biology My Passion. Thank you for watching.